we've done a bunch of updates to a lot of different pieces of software, and Susan's going to talk about them. We've been working on a brand new product that we're very excited to unveil here and show to you guys. So with that, I'd like to introduce Susan, who will give kind of a basic overview of the sort of things that we've been doing. The team has been fantastic this year. We've been really happy. So thank you.
Okay. All right. Can you hear me now? Okay. So let me start. So just to introduce myself, for those of you that don't know me, I'm Susan Ludwigsen, and I'm now overseeing the software development portion of the software development team and portion of the operation at Proteome. And so I just wanted to kind of give an overview of what we're doing and what our current philosophy is in terms of development, and kind of bring you up to date with where we are. So what we have been doing is trying to have an attitude, a process of adapting to meet the changes in the field. As everybody knows, the instruments keep getting faster, we're collecting more and more data, we're getting more and more sequence coverage and things. People are able to do much larger experiments these days, and so we're seeing scaffold files of like 14 gigs and things, and that's pretty overwhelming to the software that was not designed originally to have those kinds of loads placed on it. And with the larger experiments and things, we're trying to take a different approach and give you a more comprehensive way to analyze your results and really get biological meaning out of them. So what we've been trying to do is just take this whole approach of evolving to meet the needs of the community. Okay, so some of the things that means we need to work on are more efficient data storage. We need to get into more multi-processing to speed up performance, and we need more advanced statistical models and so forth, and tools to let you really be able to analyze these big experiments that you're all doing. So that's kind of been our goal through this year, and it will carry us into the next year too. Keep not pointing in the right place. Okay, this is a lot of text, but it's just to remind me what to say, because I was getting out 2 software releases on Friday, so I haven't had too much chance to get my thoughts together and talk to you. So besides just the software releases that we've done and the features that you're seeing, we've tried to make a lot of comprehensive changes within the company. We have a new improved support portal, and Jacob, who is going to talk to you later, who is our new support specialist, has written a lot of new documentation with FAQs and answers to specific questions that people ask, and organized it into a really nice, accessible, searchable database. So he's going to present that to you. We've spent a lot of effort on our infrastructure, which you don't necessarily see, but you see the results in the long run. We're building a much stronger common code base that's designed to facilitate better performance and multi-threading and all that kind of thing, and we're transitioning our software to that. And that should let us keep up with the pace as we're going along. And a lot of you that are probably a little frustrated right now when you're working with these giant files, this is how we're preparing to deal with that in the next generation of our software. So a lot of work has gone into that this year. We're also trying to make sure we have a much faster response when you report a bug. I think when I first started with the company like 7 years ago, there were a few bugs that didn't get fixed for 3 years or something, and I would get to them when they came out, and it would be like, oh, this person probably isn't even using the software anymore. We've tried to make sure that we have a much faster turnaround cycle on prioritizing the things that are stopping you or dealing with you. We try to make sure you at least have a workaround very quickly, if at all possible, and we try to get the bugs into the next release if we possibly can. So hopefully you're seeing the results of that. If you're not, let me know, and we will work on it some more. We're putting more emphasis on software performance. We didn't need to worry as much about that in the past, and now it's becoming really important. And so some of the improvements we've made this year are really performance enhancements. So you may not see as many new features going into some of the programs as you might have last year, but you'll appreciate that they're moving a lot faster, loading your data faster, having a more responsive GUI and things like that. And we're laying the groundwork for the next generation of products, the first of which you're going to see a demo, a preview of today. So basically, our emphasis this year is kind of a theme that you'll see through everything, where we've focused on how are you going to derive biological insight from large-scale experiments in all of our, in proteomics and in metabolomics. How are we going to deal with these larger data sets, and adapting to the changes in the search engines, and you'll see we've just put in a major new feature. What we're hearing from people a lot is that with Proteome Discoverer, with MaxQuant, with Mascot, these days they provide so many tools for pre-filtering your data, for doing your FDR analysis and things like that. And a lot of people have said, well, I don't want to have to export my whole set of PSMs into Scaffold and let it recompute. I want to see the same results so that I can compare them, or I like the results I'm getting from the search engine. So we finally are giving you that today. So you'll see. So as I said, Scaffold, we just released Scaffold 4.8. The release notes are not even on the web yet, but they will be as soon as I have a chance to write them. And I'll let you know, I'll show you some of the major things that are in it. But first I have a little quiz for you. Can anybody tell me what's wrong with this Scaffold view? There should be 3 things that maybe look strange about it. Oh, come on, you all stare at Scaffold screens all the time, I know. Any ideas? An X green line?
Oh, it's just a cluster.
Oh, that's a customized.
Yeah, that's a cluster. The blue is just that I selected one when I took the picture. Okay.
There's no p-value.
There's what?
There's no p-value, so there's no upregulation.
There's no probability, right?
Yeah.
There's no protein probability. Very good. You always see a protein probability column here, right? You usually see molecular weights too, but I didn't load it with the right database. So. But there's no protein probability column. Also, you've probably never seen this before. None and none. That must look a little weird, right? It always has 95% or 1% FDR or something. You've never seen none. And is something missing over here? No statistics view. Okay, so this was loaded with our new pre-filtered option, pre-filtered mode. And that's what I was telling you about. I've been hearing for several years, I just want to turn off Scaffold. I want it to look like Scaffold, but I don't want it to do the probability calculations. I want to be able to filter first and see everything the same as in my search engine. So that's what we have put, that's one of the features we've put into 4.8. So you can go ahead and apply an FDR or run Percolator or whatever you want to do through your search engine. You don't have to do this. This is an option, but you can. And then Scaffold will not run a probability model. So you should see all the same spectra and peptides that you saw. I will tell you that Scaffold still performs its own protein inference and grouping. So you could see some differences because of that. It's possible that the peptides might get assigned to slightly different proteins from time to time, but it's probably not going to happen that much. You're going to see pretty comparable results. So there are a few limitations to using this. Oh, well, first let me just show you. This is how you actually do it. So in the loading wizard, you'll see where you usually select either LFDR or peptide profit with high mass accuracy. there's now an extra option, which is load pre-filtered mode. And so if you select that, then automatically it's going to apply this no threshold, because what it wants to do is load everything that everything that's in your files. So that's the default threshold for pre-filtered mode. There are some limitations because we're not doing probabilities. Probabilities are how Scaffold collates spectra when you search with different search engines and load files from Mascot and Sequest or whatever, and then you want the program to put them together. To do that, it needs to do peptide probability. So you can't do that in pre-filtered mode. You have to load from a single source. You can load multiple files, but they have to be from a single source. If you did run like Mascot and Sequest within Proteome Discoverer, and they're in the same file, it will be okay, I think. Pretty sure. I should check that, but I think it will be okay. But you can't expect it to combine. If you want to do that, you need to run a probability mode. You won't see probabilities in the samples view, as we just saw. You won't see probabilities in reports, either peptide or protein. You can't run condensed during loading because we want all the data to pass through. And you will not be able to do FDR thresholding in Scaffold. It's assuming that you've already thresholded your data. But you can use a custom threshold and just basing it not on probabilities, but on search engine scores. So if you have exported everything from Mascot and then you go back and say, I only want to see the really high scoring ones from Mascot, or the ones that exceed the identity threshold by a certain amount, you can still do that. And in fact, that's one of the other major features that we put in this release, is an expanded interface and capabilities for doing the custom peptide thresholds. So the two features kind of go hand in hand. Some of you may not ever have discovered that Scaffold did allow you to set custom peptide thresholds for search engines. It only did it for a subset of search engines. Now it's expanded to pretty much everything that we support, and it's in an easier format and dialogue. So what you'll do is, in the edit menu, you'll pick edit peptide thresholds. It will come up with this dialogue. You can say new threshold, and then you have this nice form that you can choose the different types of rules, and you can choose search engine scores. If we wanted to use this for pre-filtered, we would click the little trash can on the probability, because it won't let you apply it to a pre-filtered file if it's a probability. So we would get rid of that, and we would just use Mascot. But you can see that it basically has all of the search engines that Scaffold supports as options for setting custom thresholds now. So that's another major feature. Okay, the other thing, as I said, one of our big themes this year is how are we going to deal with large-scale data processing. And one solution that we think is really could be a viable solution for a lot of people that may not have tried it yet is using Scaffold Batch. And with Scaffold Batch, you can run multiple instances of Scaffold at the same time, which you can't do with the interface. You could set up your jobs and then run them at night, or when the computer's not busy, you could have it all set up and queued, and then you just run it, which is nice. You're not tying up your computer all day. It's more efficient for large jobs because in the interface form, it has to be updating a lot of that information to build the interface. In Batch, it can just go ahead and do all the processing straight through without having to worry about that. It's faster and more efficient. It still can produce an SF3 file, just like any Scaffold run, which can be opened in the interface. You can then do any analysis you want on it, any follow-up, apply Go terms, anything you want to do, which you can do with Batch too. And it also works with the viewer. It's just a regular Scaffold file. It doesn't matter whether it was created through Batch or the interface. The other thing that you can do with Batch that a lot of people find useful is to build it into a processing pipeline, because you can run Scaffold Batch from a command line. You can put it into a script and run it as part of a whole series of steps. But what we were finding is that was really good for a few people, but a lot of people that we would say, well, maybe you should try Scaffold Batch because you're doing lots of data. They would say, well, I'm not really comfortable with writing scripts or editing XML files, or things like that. So what we have given now is if you have a batch license with your Scaffold, if you buy a Scaffold Batch license, which is an upgrade to Scaffold, you get the fully functional interface version and you also get Batch. If you have that, when you open the interface version, the regular Scaffold, you're going to see now an extra little icon. See that little gear icon on the toolbar? That is to open up a dialog to let you set up your Scaffold Batch jobs without having to edit XML or write any scripts or anything like that. And so when you do that, it first brings up a little queue, which I'll show you in a minute. And then it will take you, when you say you want to create a new one, it leads you to this form. And you can see it's set up as steps in a workflow. The ones in pink require attention. The others you could get by with just leaving them as defaults and still make a job, but you can set them. So you basically have to fill out this little form, and then you click create, and it automatically creates what we call the Scaffml, the XML file that Scaffold that holds all the parameters for Scaffold Batch. And it's all ready for you to run. And then it opens a job queue, and it will put your job onto that queue when you create the Scaffml. And then you can select it and click run when you want it to run. And you can see in this case, I was running 3 different jobs. Well, one completed by the time I took the picture, but I was running 3 different jobs at the same time. You can do that with Scaffold Batch. So hopefully this will be a good solution for some of you that are kind of getting bogged down with the quantity of data or the number of analyses that you have to run with Scaffold. So, okay, so on to the other products. Scaffold PTM was also just released on Friday, version 3.1. It has a new proteins view, which you can see here. You have options for how you want to display the coverage and the different samples in the protein sequence. This is called a stacked view. There's an overlay view and a sequence coverage view, so those are all options. You can see it has a new sequence coverage pane up in the corner that lets you see very easily where you have, and these are for modification sites, so you can see where you have them in the different samples. And then it still has the traditional spectrum and I always call them bar charts. the spectrum at the different peak depths, where you can examine the process of the A-score calculations and everything. So you still have all that information to drill in, but now you have a much nicer display of the sequence coverage or the relative, the modification in the different samples, all displayed at once and in a clearer way. We've improved some of the statistics for quantitative analysis of PTMs, and what else? Oh, I don't know if I put a picture. I don't think I did. In the quantitative view, we are displaying like the number of modified peptides over the total number of peptides as an option if you want to look at that for your PTM quantitation. We would like to do some more improvements to PTM, and I am looking for people who are interested in that to come talk to me so I can discuss some things with them, because I kind of need some input from users for some of the other ideas we have. So if you're a person who does PTM analysis, please, please, please stop by the booth and talk to me. We're in booth 218. Or give somebody your email and say I'm willing to help with PTM. Okay? All right, Scaffold Perspectives. This was not just released, but we did a lot of work on Scaffold Perspectives. Now, this Perspectives follows along with our same theme of analyzing more and larger data sets and giving more biological meaning, because the whole idea is that you can use it as a follow-up to Scaffold, where you can load multiple data, multiple files from multiple Scaffold experiments. You can categorize them. Oh, I don't have the organized view here, but you'll see more about this. Phil is going to give you a little talk about what this capability gives to you in terms of doing exploratory data analysis. But before this release, Perspectives, it allowed you to combine multiple experiments, but it didn't really give you a lot of added value over a regular Scaffold analysis. Now it does. I've been using it for some exploratory data analysis, and I think it's really cool. It does PCA. It does, it has now like much better support for Go terms on the order of the type of things you have in Scaffold. But what's really nice here is Scaffold has that very limited hierarchy where it has the MS sample, the bio sample, the category. And once you load them, you can change categories, but you really can't change anything else. It's all flexible here, and you can have as many layers as you want, and you can roll up your results to any level and do comparisons by like Go annotation, Go terms. So you can see at whatever comparison level you have in your experiment, you can see if there's differential expression of certain biologically relevant Go terms and things like that. So I think that and PCA and volcano plots and some other nice quantitative statistical tools make it now a really good follow-up analysis, especially if you have any relatively complex experimental design. But you'll hear more about that from Phil. Okay, let's see. Q plus and Q plus S. Last year was all about Q plus and Q plus S. So right before ASMS last year, we did a major overhaul of those programs. So this year, we've just kind of been refining what we put in last year. The big improvement this year for Q plus and Q plus S is that you have more access to the Scaffold filtering capabilities, including being able to actually filter on stars. And if anyone hasn't discovered stars filtering in Scaffold, it can give you a lot of power because you can make 4 different groups and you can do all kinds of filtering and sorting and things based on those groups. And now you can use that power in Q plus or Q plus S. And then finally, our newest released product is Elements for Metabolomics, and we talked a lot about that last year as well. We did a really major enhancement of that program this year. And one thing we gave it was all of this capability that I was just talking about, and which Phil will also talk about with PCA and those kinds of analyses. And the other is that we added the ability to make personal spectrum libraries. And we think that that is a really powerful tool in a metabolomics tool. So I'm going to ask Phil to talk a little bit about the personal spectrum library feature in Elements, because we think that's a really important thing that's been happening this year. So is he? There he is. I couldn't see you back there. So he's going to take a few minutes and talk about that. Sure.
Hello everyone, my name is Phil Seitzer. I'm the, oh, it's not on. Hello?
Did I accidentally turn it off? Because it was on when I started. You could hear me, right? Okay.
Okay.
When I moved it down.
I'll just try to project. All right, so yeah, my name is Phil Seitzer. I'm a bioinformatics scientist and developer, and I'm very excited to tell you about some exciting improvements to our metabolomics products in the avenue of spectral libraries. Okay, so first, those of you who have used our product maybe, and even if you haven't, you're maybe familiar with NIST, which puts out a series of very nice spectral libraries. And this year, they are releasing a new version of their spectral libraries. And we are bundling the newest version in with our metabolomics tool. which we will make available once. So the official release date is during this conference, so once they release it, we'll release it. But basically, this new library has many more metabolites and also many more experimental records. So they gather many more MS2 spectra under many more conditions. So in the new personal library tool that we've added to our metabolomics tool, you can do things, you have much more control over the spectral libraries that you search. So you can do things like take several spectral libraries, make a subset, and combine them together. So, you know, many of these publicly available libraries are very large. You might only be interested in a certain class of compounds or some subset, some meaningful subset. So we have a whole nice tool for doing that. And then in addition to being able to combine, take pieces from the libraries of your choice, you can also use your experimental results and build that into your libraries. And that has some advantages. So depending on your specific instrument, the collision energy that you run at, other technical details, this will allow you to have more accurate MS2 spectra. We also have added the ability to match based on retention time as well as M/Z. And we had intentionally avoided that before because retention time is something that is, it varies a lot depending on things like your column type and your gradient length and temperature. There's so many factors. But with this tool, you can gather spectra with your personal lab chromatographic system setup and then research, and it should be consistent. So we offer that ability now, and with this tool, we also have a whole kind of new treatment of unknown metabolites as well. All right, so an exciting workflow that this tool enables is that you can, and actually this workflow is going to come up again later in the talk, so keep your eyes peeled. But basically, we introduced this new idea. You have standards data. You can do a traditional element search and identify metabolites, search against a brand new NIST library or the libraries of your choice. But then you can take that further and parse through those results and create your own personal spectrum library out of those results. And then once you've done that, you can take your experimental data, which might be messier, and you can search against this new personal library that you just created in this step and get a much more accurate, refined result than if you had just searched your data directly against a library like NIST. So here, I just want to show the GUI a little bit. All right, there we go. So we have a lot of control about how you import your experimental results into a spectral library. So you can do it spectrum by spectrum and have a great deal of control and manual verification and editing, or you can import a large batch of libraries. And in so doing, there are options about how you want to import your experimental results into a library. Take all spectra, take high-scoring spectra, so on and so forth. So here's an example of the tool in action where I've selected a number of metabolites that I'm interested in, want to add them to a library, and then we have tools that allow you to tune the spectra so you can do things like preview them before you add them in, refine them, delete things, add things, edit things. So there's a lot of control given to you about how you want to build your spectral library search your data. So I mentioned earlier the power of unknowns. And in so doing, I'd like to introduce there's an important distinction with unknown metabolites. So first of all, there are known metabolites, such as this one right here, where we have a chemical formula, and we have a name, and we have identifiers, and we have a lot of information. And then there are things that we don't know much about. We've identified a feature at some M/Z and retention time. This is what I would call an unknown unknown, because we don't know much about it. But using our new tool, what you can do is find an unknown metabolite, save it into a spectral library, and record the experimental data associated with that unknown, and then search again and find the same unknown in other data. And that's what I would call a known unknown. Because you know what it is in the sense of you've seen it before, but you still don't know what metabolite it is. Okay, so and one more thing that you can do with unknowns is you can save them as unknowns, or you can transform them into known metabolites. So in this case, this was an unknown metabolite, and I decide that you would like to, that you think you know what it is, and you identify the major spectral peaks, and then you can input a formula and a precursor type, and you can cross-reference this and sort of do the verification on your own, and actually import this previously unknown data as known metabolite. Okay, so I'm going to give it back to Susan to wrap up this talk.
So basically, that's just a quick summary of what we've done this year. Does anybody have any questions about any of it? We're going to have a question and answer session at the end of the session where you can ask any questions you want or talk to us about the software. And so if you want to see anything about any of the new features, then we can demo it and show you how they work. Okay, so that concludes the review of the year at Proteome Software, and we're going to go into doing some actual talks, some more scientific talks. And actually, Phil is going to come back up and give you the first of the talks. So I'm just going to grab up there. Sure.
That's a very long desk.
All right, everyone. So today I'll be giving the most interesting software presentation in the world. I hope I can live up to what I've set out for myself. So Susan was mentioning that we have observed this trend in metabolomics, proteomics, where we have more and more data, more samples, more experiments. And today I want to focus on a tool that we've developed to help deal with more complicated experiments, where you have many more samples and many more variables. So the first thing is, in these experiments, there's often a great deal of metadata associated with your experiment. So you might know things like, well, we were looking at, for example, we wanted to investigate young versus old as an age group. We injected all of our samples in triplicate, so we have all these different injection numbers. Maybe we fractionated into different, we cut up the gel, we fractionated into different bands. And the point of this slide is just to show you some of the variables you might have at play in your experiment. So I'd like to use the term attribute group, which is roughly analogous to in Scaffold a category. So a kind of a thing, if you will. And then the attributes are the examples of that thing. So within the attribute group of age group, you might have the attributes of young or old. So a sample can either be young or old, or it might not have that attribute at all if it was, for example, a QC sample. So imagining that you have this, and usually people know this about their experiment, right? You know what your questions are, you know what the variables are you're interested in, and you know which samples correspond to which. So imagine taking that information and organizing it into a hierarchy. And in this hierarchy, you want to summarize information up from the lowest level up towards the higher levels. So down at the bottom, you'll have things, well, at a low level, you'll have the technical replicate level, which would be below the biological replicate level, which would be below the level at which you'd like to do your comparisons. And of course, you can have things that are even below the technical replicate level, for example, if you have fractions. And you might be comparing many different variables simultaneously, so this tree might continue. So so imagine this idea, and built into this idea, there are some specific considerations related to how quantitative values are rolled up as we go up the tree, and how normalization is performed. And I think that to really show how this tool works, I'm just going to jump into some examples, and keep this in mind as I show these examples. All right, so the way that we've implemented this in for both proteomics and metabolomics data. And so you can observe this in elements for metabolomics, scaffold perspectives, and a key principle is that it should be flexible. So when you have a large experiment with many variables, it may be the case that there are multiple designs that make sense. So you might want the ability to very quickly refine your design, or sort of see, well, what happens if we are going to look at this variable or these two variables in combination, and so on. There's just a little snippet of the GUI, where in this case this is our current working experimental design, where here we have levels, and we are designating the key points of where we're comparing our biological replicate level and technical replicate level, and then we might have unused levels in one particular experimental design, and that's okay. Okay, so first off, I'd like to talk about a fascinating metabolomics experiment that explored nitrogen starvation in fission yeast. So fission yeast is a variety of yeast. It is not the more commonly baker's yeast or Saccharomyces, but rather this is another commercially important yeast used for brewing. Here is some kombucha that was created with some fission yeast. And so the work Sajiki et al. Were very interested to look at the effect of nitrogen starvation in these fission yeast. And so they had some prior knowledge about what metabolites might be affected during nitrogen starvation. So they had a sort of a short list of some metabolites that they were going to pursue and look at the quantitative differences. They examined how nitrogen starvation, how the metabolome changed over time following nitrogen starvation. And so we took this data, and in this case, the experiment was fairly straightforward. We had basically only one variable, which was time. We had a number of replicates, and actually in this experiment, they ran samples in both positive and negative ionization mode, so we had some semblance of a fraction. And so we took this information, we designed our experiment as such, and we made a few adjustments for our run. One of the most significant is that, well, rather than searching against a small library of 75 standards, we actually searched against the entire NIST library, just to see what other metabolites we might find. And we found a very encouraging result that we were able to essentially reproduce their PCA plot. So on the left, this is the PCA plot that was reported in the original publication, and this is our brand new PCA tools result. And then, as you can see, they're quite similar, where we see this pattern in both PC1 and PC2. So that's encouraging. And we also, so we have this validation that we're more or less finding things in approximately the right quantitative profile. And just to show you, here's an example of a metabolite that they reported and then we discovered again, that actually were the individual quantitative values for this metabolite in these attributes, if you will, at these different time points, varies according to PCA. And then we found a new metabolite, guanosine, that shows the inverse profile. And this makes a lot of sense because nitrogen starvation is known to be associated with curing metabolism. So it's not really a surprise that we would observe this kind of pattern for guanosine. All right, so in this example, we were more or less able to reproduce an existing study, maybe find a few more things, but the real power of the tool is when you have experiments that are complicated, that have many variables, and especially in the case where there isn't a straightforward conclusion. So there was this study that we found where a group, Downs et al., was looking at allergenic walnut proteins, and she wanted to look at, her and her group wanted to look at the combined effects of temperature, roasting time, extraction, and the extraction method, which was, they explored different, so that's, what I mean by that is how they prepared their samples. So they were looking at solubility effects in their preparation. And in that study, they reported some things, but they didn't really come to a strong conclusion about saying that one effect was really dominant. So we started from that overlay of their experiment, and our first, whoops, wrong way. So if you just include everything in one experimental design, this is the PCA plot that we produce, which is a bit of a jumble where I don't know if there's really a clear interpretation to be had from this. And so this highlights the flexibility of our approach where, okay, so we tried this out, we didn't really see anything that jumps out as meaningful, so let's try something else. And when we try just looking at extraction method by itself, just asking kind of one variable at a time, one question at a time, then we can see that in this case, extraction method does have a significant effect, which makes sense that it would, because proteins, you have different solubility for different proteins. All right, so just to follow up on that, how should we treat extraction method? Because you can imagine to be a variable, you can also sort of imagine it like a fractionation, where some proteins will not really extract at all under one and will under another. So we explored looking at temperature and roast time in turn, treating extraction method differently in each case. And what we found was that more or less the same basic PCA structure, but we see more separation when we treat extraction method as a fractionation. So these suggest that perhaps both temperature and roast time are important separately, which also makes sense and is consistent with previously known results about the effects of temperature and roast time on these walnut proteins. Okay, so what's really exciting is when we treat extraction method as such, instead of being a variable, again as a fractionation, now our PCA we start to see more resolution in our categories. So this category here is the highest temperature and the longest roast time. This is the longest roast time, and this, the asterisk, is the highest temperature. So what this suggests to me is that there is a dependent effect on temperature and roast time, which makes sense. For those of you who have ever cooked food in the oven, if you cook something for a long amount of time at a low temperature, or at a short amount of time at a high temperature, provided that you have significant conduction of the heat through the food, you'll see approximately equal effects. So basically, what I believe these results show is that both temperature and roast time are dependent on each other in the effect of the quantities of these allergenic walnut proteins. So just to show you side by side, the dramatic effect that experimental design can have in the interpretation, and it's very easy, very quick to do this. All we had to do to go from this to this is just move this down, which our GUI allows you to do very quickly. Okay, so of course, this approach does have its limitations. There are many other statistical designs. So for example, if you have a block design with nuisance variables, which we offer in Q+, by the way, you might be able to see more power in this approach. There are other models, the linear mixed effects model, and I'm sure that statisticians who know of many more models, you can frame, that. And of course, we can do more. And then when you're thinking about normalization, you might want to have multiple baselines or a more complex normalization scheme than the one we offer. But that said, these are exciting directions we hope to address in the future. And I believe the tool is quite powerful and useful for what it can do. And it's available now, and it's currently released in Scaffold Perspectives and Elements from Metabolomics. If you'd like to learn more about this or come talk to me, please see me at Poster Thursday 342 and at any time at booth 218. Wrong way. Okay, I'd like to acknowledge the whole team, and especially for Melanie Downs, who very graciously provided some information to help us reproduce her results in relation to the walnuts data set. Thank you very much. Does anyone have any questions? Okay, no questions. Okay.
Am I next? Okay. Okay, so we've been doing a ton of work over this last year, and you've seen the fruits of a bunch of it. Some of it has been under the covers, which has been sort of like the manipulations of the data structures to make things for the future, to move faster. But there's also been a lot of work in terms of new graphical elements and new statistical analyses methods. But we've also been hard at work over the last 6 months or a year trying to wrap our brain around a new data acquisition strategy, which we feel is going to be extremely important over the next several years. Not to replace DDA and DDA methods, but to augment DDA methods. And so and I'm very excited to say, we're going to show a demo of this software that we've been building. Seth is going to do that, and he's going to also talk about the actual program itself. But before we go through that, I want to talk a little bit about sort of the basics for data independent acquisition, to kind of give you guys a framework for how to interpret this piece of software that we've built and what kinds of things you can do with it that are unique, particularly in the realm of quantitation, which is where we're specifically excited. So the difference between DDA and DIA is fairly easy to conceptualize using this framework, where say you're going into a library and you ask the question, well, I'm interested in checking out Harry Potter. How do I find out where Harry Potter is? And if you were to think of this from a DDA perspective, you'd pull a bunch of books off the shelf and put them into a cart, and then you'd look down the list, you'd say, okay, well, I found Moby Dick, I found Pride and Prejudice, I found War and Peace, but I didn't see any Harry Potter here. So is it there? I don't really know, actually. And a better way, like the most logical way of answering this question, would be to go look up in the card catalog and say, Harry Potter should be in the J.K. Rowling section, and you should go look over there specifically on the shelf, okay? This sort of strategy for pulling a book off the shelf based off of its card catalog index is really analogous to a data acquisition strategy called PRMs, where you're targeting a specific peptide, you know exactly where it should be in mass and retention time space, and you acquire spectra to pick out data, experimental evidence for that peptide being present or absent, okay? So the question that DIA tries to answer is, what if you wanted to apply this idea of using a card catalog, but across an entire genome, right? Is it possible to do targeting of every single peptide in an experimental condition? And so the answer is, you can, but with some compromises. And so I'll tell you about how you can do that and some of the compromises you have to make. Specifically, that there are multiple ways of doing this sort of data acquisition, and that there are some caveats to doing this sort of data acquisition, but we feel that in many experiments, the benefits of DIA actually outweigh some of the consequences. Particularly for quantitative experiments, we're interested in quantitative measurements from group to group, from sample to sample, across many different samples, right? Many different biological replicants. So, as I mentioned before, with PRM experiments, you end up acquiring fairly narrow precursor isolation windows to target a specific peptide of interest. And you do your quantitation using fragmentation spectra, where you acquire a chromatogram over time using that evidence, right? With DDA, on the other hand, you again use a narrow isolation window to say this is definitely what peptide I'm looking for, but you sort of sample them in a data-dependent manner where you collect a sequencing spectrum for every single peptide that you can interpret, but only one sequencing spectrum just to confirm the location. Consequently, you have to do your quantitation with the precursor signal in most cases, or in a single scan if you're using iTrack or TMP, okay? So DIA represents something of a compromise between these two different approaches, where you gain in that instead of with PRM, which is only targeting very specific peptides, you can analyze an entire proteome, or nearly an entire proteome. But you get the selective quantitation of being able to measure fragment ion scans for your quantitative measurement repeatedly over time, and again being able to look at chromatograms. Okay, so what do these compromises kind of look like? So from the standpoint of the experimental constraints, you can only scan so fast without sacrificing your quantitative measurement. You need to be able to measure at least 6 to 8 peaks, or 6 to 8 points across a peak to be able to say, I understand the shape of this peak, to be able to measure it quantitatively. And so you get that constraint measured up against the speed of the instrument, which might be between 10 and 20 hertz. So the compromises that you have to make this set of constraints work is that first you have to accept a higher level of interference by widening a precursor window selection. So instead of with DDA, you might be looking at plus or minus 1.5 MZ in terms of precursor isolation. With DIA, you might be looking at plus or minus 10 MZ wide isolation, okay? And the other thing is that you need to limit the measurements you make to an area of interest. So instead of being able to look at all precursor peaks from 400 to 1600 MZ, typically we make choices to limit that to 500 to 900 or 400 to 1000, where the majority of informational peptides actually live. Okay, so what does this data acquisition strategy look like? Usually, again, you'll scan from 400 to 1000, you'll collect a precursor scan, which is just sort of for information purposes to kind of give you an idea as to the landscape of the proteome. And you'll collect a fragment scan, and then another fragment scan, and tile these across the entire window space. In this particular case, I'm collecting 24 MZ wide windows here to scan from 400 to 1000 MZ. And then I'll go and collect another precursor scan and repeat the entire process, okay? So, and usually I try to tune this so my distance between scans is somewhere between 2 and 3 seconds per scan. So if I collapse that up into one layer, that would look like this. So let's see exactly what's going on with one particular scan. So for example, for measuring this particular peptide, this VLENT peptide, that would exist in this narrow mass range of 784 to 808 MZ, okay? And if you were to look at a spectrum representing that peptide, you would get something that looks sort of like this, where you get fragment ions across the MZ space that signify that that peptide is present. But one of the biggest advantages of DIA is that you actually collect these repeated measurements over and over every 2.5 seconds. And now, because of that, you can actually start thinking about quantifying on these fragment ions, where you start to measure over retention time space these different shapes of these chromatograms for these fragment ions as representative of the quantitative measurement. Not only do you make, instead of making one measurement per peptide at the precursor level, now you can make n different measurements where n different fragmentation ions are being measured. Okay? So again, if you're fragmenting this peptide, you would end up seeing right here a DDA style spectrum. This would be a DIA style spectrum, where you're measuring scan after scan after scan. And if you were to look at it down this axis, you'd be seeing what we call a chromatogram measurement to signify the detection of that peptide. So most of the data, instead of showing you spectra that look like this, I'm mostly going to show you chromatograms that look like that. Okay, so there are some caveats with these types of measurements. The first one is that the ions that you're interested for each individual peptide, while you can see them rise and fall over time, retention time, they're not necessarily centered in the middle of the window, of the precursor isolation window. This has two effects. One is that, particularly that because you are making a measurement that's for an entire window, the collision energy that is tuned by the instrument is not necessarily tuned specifically for the peptide that you're measuring. The collision energy will be tuned for the middle of the window instead. The second is that usually in every set of scans you acquire multiple peptides at the same time. So you end up seeing what appear to be fairly noisy spectra, but that's simply because you're fragmenting 2, 3, 4, sometimes as many as 20 different peptides at the same time, and needing to make quantitative measurements on all of them at the same time. And that the third one is that with certain types of instruments, there can be some edge effects that cause suppression of signal. So for example, if we look across this 784 to 808 range, instead of thinking of quadrupole transmission efficiency as a square wave where you would say, okay, the ions get transmitted equally well all the way across this range, it's usually kind of trapezoidal, and you end up not collecting the most intense ions for the peptides that elute on these wings. So for example, the signal for the red guy, for example, will be slightly lower than he would be if you were to actually make that measurement using DDA, or PRMs, really. That said, these sort of parameters are consistent from experiment to experiment, run to run. So these don't affect, they're more like global effects rather than they're effects that change one sample to another. Okay, so if I wanted to set up a DIA experiment, what kind of things would I need to consider? And the first one is this windowing range. So why are we measuring 400 to 1,000 MZ? And that's really a result of graphs like this, for example, where I'm measuring, this is the precursor MZ over here, and the number of peptides that I can ID across that range. And so if I look again at this other measurement, this is another sort of way of thinking about that. This is the precursor TIC across for every one of those different bins, okay? And so it makes sense based on this chart to start to think about making a measurement from 400 to 1,000 MZ, because that acquires data on the majority of peptides that you can see in this sort of window. But in some experiments, you might be more interested in collecting more targeted data where you get better precursor isolation, but it's from a narrower range. So oftentimes people will start to select between 500 to 900, or between 450 to 850. Okay, and so another thing to think about is why 24 MZ wide windows, or why wide windows in general. One of the sort of surprising things that we can see is that, that if we're analyzing data that is effectively like PRM data, we can see it across, in this particular case, across 60,000 peptides or something of that order, that 90% of them we can get quantitative ions, at least 3 quantitative ions per peptide. And that we can't always guarantee that for these 24 MZ wide windows, but again, this is kind of another compromise where as long as we can quantify the vast majority of peptides in our experiment, we're fairly strong. So one important characteristic that you need to make in designing these types of experiments is to know how fast your instrument is running and exactly what sort of data that you might be trying to acquire, and tuning the experiment to that specific set of parameters. So for example, if your instrument can acquire something like, if you're wanting to achieve a duty cycle of 2.5 seconds or that sort of thing, you can calculate based off of the mass to charge range that you need exactly how wide your isolation width needs to be. And so while we don't need to go through the math here, if you want these sort of slides afterwards, I'm happy to give them to you later. Okay. So this is one style of acquisition here, where we've got 24 wide windows tiling across 25 windows of 24 MZ wide size. So this is just one example, but there are lots of different ways in which you can tune that. So that style of experiment is very analogous to the sort of original swath approach in which they were doing fairly wide windows across an entire range. And the reason why is then you can get an entire quantitative experiment done in one set of samples. So that's what we call these wide window DIA experiments. We have a different style so we actually end up supporting using this tool that we are going to demo here. several different data acquisition strategies. One of them is going to be gas phase fractionated narrow window DIA, and I will show you exactly what I mean by that in just a second. And the second one is these overlapping windows, which I'll show you an example of what that means too. So these are two different sort of additional methods that you can use to get more detections or more peptide signatures out of your data set. And then a third, which is becoming more and more popular, are these variable width window sizes that have been proposed in like the SWATH 2.0 approach. And so those are additional experiments that we're supporting. So one of the approaches that we will try to support coming soon is DIA with a specified windowing margin. And then there are these additional approaches that are currently not supported, but if there's a lot of interest from the audience, we will start looking into these, specifically multiplexed MSX, multiplex DIA, and MSE, or ion mobility-based MSE. So in terms of these other alternate acquisition strategies, the first one, gas phase fractionation, we find extremely powerful. And Seth is going to talk to you later about a workflow inside this program that I think is extremely important for how to interpret this type of data. Okay, so with the normal sort of 24 wide window DIA, one run per quantitative sample, you end up acquiring data that sort of looks like this. But if you had enough sample that you could burn and enough sample time, so enough instrument time, where you could burn a second acquisition, you could end up acquiring two different DIA experiments. One DIA experiment where you're looking from 400 to 700, and a second DIA experiment where you're looking from 700 to 1,000. And here you'd be able to narrow down the window size to accomplish a 12 MZ wide window, which is going to give you a lot more power in doing data analysis afterwards. Okay, so what if we were to extend this idea even further? So instead of looking at one scan divided into two, what if we were to look at one scan divided into 6? So instead of looking from 400 to 700, we look from 400 to 500 with 4 MZ wide windows. Now we start being in the range of being able to think of this as targeted DIA, where you're essentially acquiring PRMs across an entire experiment for an entire sample, where you can target every single peptide in that experiment with the same kind of precision that you would get with the PRM. This is extremely powerful when you do an experiment like this. You end up having burned through 6 different acquisitions for your experimental analysis, but your ability to make detections and quantitation out of this type of experiment is unprecedented. It's extremely powerful. And so one of the strategies that we built into this new software is a new type of workflow where you end up acquiring your quantitative samples using this wide window data format, where those are in 24 wide windows, one acquisition per quantitative experiment. And you end up taking a pool of every one of those samples, at least within one replicate, to have in your experiment a representation of every peptide that you might see in your experiment, pooled together, and then acquire these narrow window data, right? Where you're essentially doing PRMs across an entire experiment. You make your detections in that data set, and then you quantify those detected peptides in your wide window data. This gets us up to levels that are fairly unprecedented in terms of our ability to make detections out of this type of data set. So with a fairly simple tweak to any kind of DIA experiment that you've been doing or considered doing in the past, we think that this is really an important workflow. And we built this in from the ground up for you to be able to do this starting from day one with the software. So I mentioned overlapping windows. This is an additional approach that we think is really powerful, and we built it into the software to be able to manage this type of data. So in terms of taking these scans that are abutted one next to the other, and then collecting a precursor scan and another batch like that. We're talking about, across those windows, just shifting over every other scan and acquiring another one. So every other scan is shifted off from one to the next, okay? And so here, instead of acquiring complicated spectra that look like this, we end up acquiring spectra overlapping where we can start to deconvolute this data on the fly. So with this simple quantitative method, we can actually extract out much deeper information. And let me show you how we can do that. So for example, here, this peak is present in both of the previous windows that have been offset and both of the next windows that have been offset. So for that one, we say, well, that's a common peptide, or the common peak, we should separate that out. But there are going to be some peaks that are only in this lower region, in this green region, okay? And then there are going to be some peaks that are only going to be in this blue region. And then some peaks that are just noise. And so we can take this fairly complicated spectrum and just by looking at the previous and next duty cycles, be able to make much more simple spectra that we can make detections. This really simple tweak in terms of your data acquisition strategy gets you a twofold isolation. And so now instead of thinking about 24 MZ wide windows, we can still acquire 24 MZ wide windows, but when we overlap them, we effectively get a 12 MZ isolation using this algorithmic trick. So this is another trick that we built into the software from the ground up, again, from ground zero, from day zero. Okay. So I want to take a few minutes and show you what this data sort of looks like and give you a hint for the power of the approach that we're showing and the software that we've got. So this would be, for example, a set of chromatograms for all of the fragment ions associated with this superoxide dismutase peptide, okay? And spanning across a long gradient, a 2-hour wide acquisition, okay? And so this right here is where the peptide is, and if I were to zoom in, I actually get this really beautiful chromatogram that shows a representation of what that peptide is. You wouldn't necessarily be able to pick out this peak from all of these others unless you had some kind of a software, but many pieces of software are actually fairly good at this. If you were to interpret this with Skyline or something like that, this is actually a very doable type of analysis with Skyline. Okay, now let's look at this other peptide from superoxide dismutase. This is a +3 peptide, which has a much more complicated chromatogram up here. And let me show you, this is where the peptide actually is. But this is where the acquisition is. And so I want to show of hands. Do you think it's this peptide or do you think it's this peptide is the actual representation for this? So is it, what do you guys think? Is it this one here? Do you think it's that one there? And this one here? Anyone? George, maybe? Okay, well it's this one. And how do we know that? Is actually pretty cool. So using some sort of a library-based approach where we've got a library spectrum up here, we can say, okay, the highest ions that we should expect are Y8, Y9. Y8 should be slightly higher than Y9. We see that right here in our actual acquired spectrum. We should see the next highest peaks being these Y11 and Y12, and we see those over here, and we see a fairly reasonable ladder all the way across. And so we can actually make a detection of this peptide despite the fact that there are so many other ions from so many other peptides being isolated at the same time, okay? This is really powerful in terms of being able to do these detections. And this is the sort of thing that you can't get with a tool like Skyline or that sort of thing, because it's much harder to go through manually and try to say, okay, is it this one or is it that one? It's much easier to let software deal with this on its own. Okay, so we've also, and this is, I think, an important characteristic that differentiates the tool that we've built separate from other tools that exist. And that is that we've, instead of having to, with Skyline or with Spectrum Nut or something like that, where you actually have to go through and figure out for every single peptide, what are the transitions that are viable to quantify that peptide. That's an impossible task when you start to think about 40, 50,000 peptides in a single experiment, where you got to investigate every single run, every single time. So we've developed this fairly sophisticated algorithm that does transition refinement for you automatically. So it picks out these peptides, these particular peaks, these ones in green, and says, those are the ions that are good for quantifying this particular peptide. This one in yellow, useful for detection purposes, but not necessarily useful for quantitation. And these red guys, they're not useful for anything. They're just essentially interfered with by everything, and they're probably low mass ions. And so we can make that analysis of this particular peptide, say this is the actual shape of the peak that we've got here, and make our quantitation measurements off of just those uninterfered ions. And this becomes really powerful because then instead of having to click through every single spectrum, every single scan, and try to interpret this data, a lot of this has been now automated such that you don't have to. We've made it so that you can see this information, but you don't actually have to do it. And that, I think, is the last sort of stepping stone that is required to bring DIA from being just a targeted sort of strategy that is interesting but not necessarily a useful strategy for core facilities or individual labs that are interested in doing this type of experiment. This is now a strategy that I think is viable for doing with core facilities, doing on all of the experiments you've got, where you've got tons and tons of samples, tens of thousands of peptides, where before you wouldn't be able to do this kind of analysis without tons of manual intervention. Okay, so there are lots of caveats here that I proposed, but there are some really nice things that we've got in our favor versus doing this with the targeted approach. So if you were to generate like targeted SRMs or PRMs, you'd have to generate a hypothesis, you'd have to pick peptides and do retention time scheduling and then transition refinement all ahead of time before you start acquiring your data, before you start deciding what peptides and transitions I want to look at, which is extremely time consuming, very difficult. With DIA, you end up essentially moving your data acquisition to the front. You don't have to think about it, you just acquire the data. And then afterwards, you do your hypothesis generation, your peptide selection, your extraction, your refinement afterwards. And what we're saying with this tool is that now all of this stuff in blue can be automated using these computer algorithms rather than something that you have to do manually by yourself. Okay? And so let me give you an idea for the power of this type of tool. Okay, here are 3 replicate acquisitions made using DDA, Search with Comet, looking at unique helopeptides, where we see about 30,000 peptides on average. What's fascinating here is that the overlap between these is okay, between these replicate acquisitions, but it's not really great. And if I start to look at this new method encyclopedia that we've got built into this new software that we're releasing here, we start looking at the idea of being able to quantify and detect 60,000 peptides instead of 30,000 peptides. Because we can use a lot more information about what peptides we're seeing, the structure of the peptides, the retention time accuracy of the peptides, all of this generated off of this narrow window data acquisition strategy. And we end up being able to make the same measurements every time. This circle here in the middle, where we make the same measurements in all 3 of these replicates, is bigger than all of this combined, right? This is extremely powerful. And not only is it powerful in terms of detections, it's really powerful in terms of quantitation. So this is some data published by a friend of mine, where she was using this tool to look at coefficient of variation on replicate runs in yeast. Okay? And of this data set, where she's looking at a 1% FDR and at least 3 transitions automatically picked for her, right, by this tool, automatically generated with no manual intervention here, she can see 90% of her data with at least a 20% CV. And that's shockingly good when you compare that to anything that you've ever seen with DDA. The median CV that we're looking at here is 7.7. Now this is a beautiful experiment because Lindsay is really good at collecting data, but this gives you an idea as to the power of this type of experiment that you cannot get with DDA-based methods, with SILAC, with iTrack, those kind of strategies where you're doing the quantitation at the MS1 level for the most part, where you have to deal with interference with iTrack or TMP at the MS2 level. This is a really powerful method. So in conclusion, this is not a strategy that works for every single experiment. We're not talking about doing away with Scaffold and DDA completely. What we're saying is this is an amazing tool to be able to augment the types of data collection that you're doing here. And we've been trying to build this software package to give you and make it so that you can leverage these new data acquisition strategies. So we think they're ripe right now. And so with that, I'm really excited about this new tool. Seth is going to show you how it works and also how it actually performs. He's going to do an actual live demo. So with that, I'm going to turn it over. Thanks, everyone. Do you guys have questions about this? Do you want to see it first? I want to see it. It's good. You want to just drag it over?
Maybe.
All right. So I'm going to talk about this new product that we're calling Scaffold DIA that is designed to work with these techniques that Brian's been talking about and the algorithms he's been developing. I'm a developer for Proteome Software. I've been there for about 5 years, and I've been working on this project for about the last 6 months. I definitely haven't been in it alone. Absolutely everyone in this photo has contributed to this project. I want to thank Susan and Phil especially for the work that they've done in helping kind of bring this vision to life. And I especially want to thank Brian. He has really put a lot of time and effort into this. He's done all the core work of these algorithms and really put a lot of time into the vision behind this product. So like Brian was saying, there's a lot of motivation behind using DIA. A lot of people are trying to look into it and kind of solve some of these problems that we're tackling here. And Brian has shown what you can do with these techniques, the unprecedented levels of quantitation, the coverage, and the actual simplicity of gathering a lot of this data. DDA, especially when you want really incredible coverage and repeatable quantitation, it becomes very complex. You need to do a lot of replication, maybe fractionation, and DIA can do a lot of this for you in a single run like Brian showed. And this saves you a lot of time, saves you a lot of money. You aren't going into the lab to gather all of these samples and acquire all these runs. And so with this all behind us, we set out to build a product that would really bring DIA into the lab and into biology in a way that it hasn't before. We really wanted to integrate these new algorithms and new approaches into a tool that does what people love about our software, does what Scaffold does. We wanted to really make it simple and easy to use and bring in a protein-centric approach to this data. This is something that really hasn't been done with DIA data yet, and we're really excited about what we're able to do with it. And we also, like Susan was addressing, we're really trying to build this as the future path for our software, building incredible levels of performance and all of the great advantages that we've kind of been putting into all of our software, the tools and the interfaces that people really like to be able to use. So we've created what we're calling Scaffold DIA. And Scaffold DIA is a one-step solution to analyzing DIA data. And it uses these library-based techniques that Brian has been developing. So what you see here is you may have collected your experiment, you have all this great data, you want to analyze it. You're going to take it, you're going to put it into Scaffold DIA. The question is, what is this library that you need to search it? So what knowledge, what do we need to know to identify peptides in our data? The classical answer is we just take a FASTA database. We can look at protein sequences that we get from genomics. We can figure out what peptides are there and figure out how they're going to fragment. And this has a lot of strengths. You can get great results with it, but it has some downsides. You know, the theory only goes so far. We're not fully recreating what's going on inside your instrument. And so things like spectral libraries that are built using true observations of what peptides do when they're in an instrument are really great for getting higher quality identifications. And things like SRM or PRM techniques make use of this. But there's more information that you can use even than just fragmentation patterns, retention time, known interferences, and what you actually observe when you run DIA is really important. So, you know, all of these sources of information have great advantages and, you know, have different trade-offs. So when we wanted to ask, what library do you need to search your data with Scaffold DIA? Our answer was all of them. We want to support anything that you have. So Scaffold DIA allows you to search your DIA data using whatever source of knowledge you have. If you just have a FASTA, you can go ahead, put that in, and get results out the end. But just the same, if you have a spectral library, be it from a DDA experiment you've done, you know, a library from NIST, some library that you've downloaded from the internet, you can put that in and use it to identify your peptides. But we also allow the use of these more detailed libraries that we're calling chromatogram libraries that contain all this full detail of what's going on in a DIA experiment. Not just the fragmentation patterns, but also the retention time of things, what interferences are going on. And we actually allow you to create these libraries out of your data and then reuse them and you know, reflect them back into your results. And this is really powerful and enables a lot of workflows that we're excited about. The way we support this huge variety of workflows is just with a couple of key algorithms that Brian's worked on for us. One of them that you guys probably haven't heard of because we haven't told anyone is called Xcordia. And this is similar to an approach called Pecan that's been explored. We're using an X-core scoring model. And it is very much a traditional FASTA-based peptide search engine for DIA data. So all you do is you have a DIA sample or many samples, you have a FASTA, and we're able to identify peptides out of it. But not only are we able to identify those peptides, but we can actually do this fragment-based quantitation. We can align samples based on retention time, and we can generate chromatogram libraries out of the results. So starting with just a sample and a FASTA, we're able to get this huge wealth of information out. And then, of course, the other algorithm we use is called Encyclopedia. Encyclopedia is more of a traditional library-based search engine, and it allows you to identify, quantitate peptides in your DIA files using a spectral library or a chromatogram library. And again, it's going to produce these peptides. It's going to do the fragment quantitation, and it's going to do the RT alignment, and you can actually produce new libraries out of it. So say you have a spectral library from somewhere, you can search your DIA data against that spectral library and then export all of the detailed information about the retention times you saw, what interferences you saw, which fragments were great for quantitation. And so this enables Scaffold DIA to use this workflow that Brian spoke to in his last slide in a single step. If you have DIA data and this narrow window pooled data, Scaffold DIA will, in a single step with no intervention from you, create a chromatogram library and then use that to very reliably identify and quantitate peptides in all of your samples. And we feel like this workflow is where the immense power of this program really comes in and shines. So with that said, I'd like to actually show you what the program looks like with some real data. Brian, do you want to talk briefly about the acquisition you did?
Yeah, sure. So to start, these approaches, Encyclopedia Xcordia, are they're open source tools that the McCoss Lab has been developing, and we're very excited about them in a way that we can roll them into this program. So they're designed, those tools were originally designed to deal with peptide-centric data, thinking of things more like Skyline and that sort of thing. Here, we're trying to build up to a much higher level, to be able to think about protein-centric data, and to be able to calculate protein-centric false discovery rates, and think about doing higher level analysis, again, like how we've been doing with elements and with perspectives, with the idea of thinking about PCAs and heat maps and volcano plots and bigger high-level approaches to doing this analysis, rather than, and honestly more scaffold-like approaches to doing this sort of analysis than you would typically get with, again, a spectrum knot or a Skyline approach to this sort of data. So with that, Seth, you want to show this off?
All right, so this is Scaffold DIA, and this is the first thing you'll see when you want to load some data into our program. Like I said, we've really tried to make it a one-step workflow so that you can set up what you want the software to do, let it go, and what you see out the other end is your results. You don't have to come in and kind of do any refinement. So we have, I don't know if anyone here uses Elements. We've kind of used the same concept of a workflow to have you set up kind of how you want to approach searching your data. We allow you to save and load workflows to files. So if you have a workflow that works really well for you, you can apply it to different experiments very easily. And what you see here is what you need to do to set up one of these pooled sample searches that Brian was talking about. You really don't need to specify a lot of parameters. The main thing is you simply put your raw files for your reference samples and for the actual quantitative samples you want. And then the program will go ahead, use the FASTA, find all the peptides it can, generate a library from it, and then identify and quantitate those peptides across all of your samples. And so once your data is loaded, we have all of these flexible organization tools, the attribute-based organization that Phil was talking to, and that allows you to have a very high-level view of this data. So this experiment here is a few samples that Brian generated, and he kind of used the techniques he was talking about in his talk. So the quantitative samples here, there are 4 samples in 2 groups, and these were acquired with the overlapping 24MZ wide window DIA. And the library we used to identify them was generated from these gas phase fractionated samples. And just the quality of these results are really incredible and really nice to see. And what Scaffold DIA does is takes all of these incredible peptide identifications and quantitations and rolls them up to the protein level and really gives you the ability to interrogate your data at a high level, but still drill down into the specifics of the identifications and what the algorithms and the instruments are doing, with your data. So the numbers we're seeing here on the right are quantitative values that are taken from these fragments, the fragment chromatograms that we've been able to extract to refine to ensure that there's no interference. And they're all rolled up not just to the protein level, but also what we're seeing here is the 2 groups of samples that are being combined together and rolled up. And so this is a fairly high level of information, but if we double click on a protein, we see that we can now immediately see all of the peptides that were identified for this protein across these samples. So the top table here shows the various peptides that were identified. And then here we see each sample where that identification was made. And so we have this great information kind of across the whole protein, what peptides were identified, where in the protein are they, and on the left here we have the sequence coverage, how it varies between samples. Although it's actually really incredible, we have such good repeatability because of these library-based techniques. We're very rarely missing peptides, even if there are large quantitative changes. You can see here the quantitative repeatability is really incredible. This is, about 0.15 log 10 units, which is essentially no change, even within this high variability group. And then the really interesting thing about this view is being able to drill down into the individual peptide identifications that are being made across your samples. So on the left here, we see the XIC for the precursor of this peptide and its various isotopes. And you can see that it's not a very clean XIC at all. There's some interference with all of the isotopes actually. But when we look at this right-hand chart, these are the fragment chromatograms for this peptide. And this, you can see that some of the fragments are interfered with. They maybe belong to this precursor that's on the shoulder of this peptide, but they're being rejected by the automatic transition refinement. So the green chromatograms here indicate the fragments that were the highest quality and used for quantitation. And then, of course, the blue are still high quality, but weren't quantitated, and were used for identification. And then, of course, this black one here has been kicked out. We don't want to consider it because it clearly shows interference. And what's really interesting here is that this shows the power of quantitating using these fragments, of collecting the fragments at many time points, because the level of interference in the precursor is so huge that if we were doing precursor quantitation, we'd really be measuring the wrong thing. We'd be measuring this more intense peptide that's interfering with it. And this interference is fairly reliably reproduced across the samples, but you can see there's much greater variability in the interference of the precursor than we're measuring in the fragments. And the fragments are very consistent and very easily identified. So it's really exciting to have this high quality data and the ability to view it at a very high level, to draw high level conclusions, but still drill down and look at the fine details of what's going on in your chromatography and inside your instrument as it's collecting this data. And like our other programs, we've built in all of these tools for very high level interrogation of what's going on, statistical analysis of the difference between samples, and tools like PCA that allow us to see very clearly that the two groups do have a clear separation in their quantitative profiles, that there are definitely differences in the proteomes of these two different treatments of the cells. So with that, are there any questions? Yes.
So far as I see, this software allows you to import directly the raw files. You don't need to convert them through max quant, so it's reading the raw files and all the intensities, and it's doing automatically log 2 of the intensity. You don't need to have a file to import already processed. So you filter the raw file.
Right now, at this point in development, we're only loading MZML files, but the idea is that you'll be able to specify, give it any raw file and it will do everything you need to do in the processing. We really want to make sure that this program is a one-stop shop, that you don't have to spend a lot of time pre-processing, clicking buttons, sitting through things, that if you have data and you have some library, you give it to the program and it will just run it. So yes.
Can you explain a little bit more about the retention time alignment? How do you set the initial retention time? And what if there are reproducibility on the retention time from day 1 to day 200?
Yeah.
And how do you set that initial one, I guess?
Yeah, so there's some advantages here in that if you generate your 6 samples, your narrow window chromatogram library data, if you run those back to back to back, there shouldn't be any retention time variability, right? Or there should be very minimal retention time variability. So now you've got detections where you've got an assigned retention time. And those detections you can make without knowing anything about retention time ahead of time. Okay? So you can say, okay, well this peptide, I think it's over here, and that's great. So you're not going to be as deep as you would otherwise, but you'll be able to make a bunch of assignments. Okay. So then we have a second pass. Well, then we go and say, okay, now that we've made these detections without knowledge of retention time, can we apply retention time to get some use out of it? And in that particular case, even if you have wide warping between the library and your actual acquisition, now we know that a bunch of those peptides are real, true anchors, and we can say those detections map the true retention time curve. And even if it's a wide curve where the tails are different from how they should be, you should still be able to make the retention time alignment prediction. And then from there, we go back and say, okay, now we know what the real retention time should be using this data-dependent approach, and then remap the retention times to their correct locations. And then we get power out of it from that. Make sense?
I think so.
Okay.
I got two questions. One is, since you guys are doing the identifications with your own algorithm now, have you done any validation against other search engines?
Yes. As I said, the algorithms that underlie Scaffold DIA are open source Macass Lab algorithms. So the whole point of that is, there are scientists outside of us that are continuing to do that work, of which I represent part of that. Okay, so that is sort of a separate development of these tools, and that gets continued, and Scaffold DIA gets all the benefit of new approaches that are added into those toolboxes that are applied. So in that analysis, there have definitely been benchmarks against things like Skyline and like DI Empire and that sort of stuff. So, but that's not, we're letting the Macass Lab people do that. Again, as I'm like a joint member between.
Yeah. So are there any restrictions with the type of data that you feed into it in terms of, in using regular Scaffold, I've had a challenge with nanoflow data sometimes because of the file sizes.
Right.
So is there going to be any type of restriction from that aspect, nanoflow versus
No.
cap or microflow and that kind of thing?
Yeah, no,
Or any file size restrictions?
no, there aren't file size restrictions. If you're using TOFs, usually the file sizes are a bit larger and the analysis takes more time, but it's all about time, it's not about, actually the slide that Seth didn't show, which you should totally show, is about how well threaded this new algorithm, not only the algorithms, but also the actual GUI for the tool is. It is shockingly fast, and it's due to a lot of work that Seth has put into it.
And it's going to be faster. He's
Yeah,
not-
so-
this is still very much under
Yeah,
development.
I will say, performance is something we really tried to build in at the very ground level to make sure that, multi-threading, that we can stream things through memory as much as possible to bring down these restrictions on file sizes, reduce the amount of resources you need. This chart here is showing what the GUI is actually fully multi-threaded now. So this experiment here was totally running on this cheap laptop and using all the cores we can, and we're able to quantitate over 5,000 proteins in this experiment in just a couple of minutes out of our results file. So we've really focused on performance as a first-class citizen in the development of this.
I have a question about quantification based on fragments.
Mm-hmm.
I'm always wondering about quantifying stuff based on fragments because imagine a situation you're measuring on a thermal instrument, you set your AGC target to 100,000 ions, and you only have one precursor evaluation. Everything's good, and if that's your like a pivot experiment that you will be quantitating relatively. And then in the next run, another ion comes into the isolation window. And now, let's say in the first run, the instrument was able to fill the trap in 10 milliseconds. The second run, there's another ion, and it's the same intensity. So now it fills the same current as the ion in 5 milliseconds, and effectively your original ion becomes 2 times smaller, sort of.
Right. So the resolution on that ion becomes 2 times smaller in terms of the intensity resolution, like the number of bits that you use to represent that ion essentially drops in half. But the actual intensities themselves get scaled by the instrument. So
they do that. I really don't know what exactly were they doing in such a case.
yeah, so yeah, you definitely need to, and this is definitely something that instrument vendors have figured out a long time ago, that you need to keep consistent ions, even if your ADC targets are somewhat different. So yeah, it's totally okay. You can get some other effects by introducing very large ions, precursor ions.
Yeah.
So for example, you can get coalescence. But in the end, we end up choosing which fragments to pick based off of all of the quantitative experiments, not just one quantitative experiment. So for example, if you get a large peak that happens to be in that window at the same time, causing coalescence on some of the fragment ions in 3 out of 6 experiments, we end up choosing a different ion instead that isn't getting shifted over. So that actually makes it a much easier method for quantifying, because when we think about quantitation, we think about all of the data together rather than just one sample at one time. Cool. All right, well,
Can I say?
yes.
Okay. I just want to make sure, because Seth did a fantastic job showing off the live demo here, I want you to understand that this is still very much under development. It's not available yet, but will be sometime later this year. And in the meantime, we are going to be meeting some people to be beta testers. We're going to be soliciting people that are interested in perhaps the software, getting feedback from you up front to make sure that we tailor it, to meet your needs and things. So if you have any interest in this, we'd really love it if you would either see one of us today or drop us an email and just let us know that you're interested in the DIA product, and then we can contact you and kind of interact with you as we develop the software.
How do you envision the DIA software versus your current scaffolding, where you have the software available for the licensing, and also if there are people who are interested in it to look at the data at the preview mode?
Oh yeah, sure.
Is that the same makeup you envision for this?
Yes,
software?
yes. That's been consistent through all of our software. It's something we strongly believe in, that that you need to be able to show this data to people. You can't just give them file like tables and Excel or whatever. You definitely need to be able to demonstrate the results to people. So it looks like a scaffold product. It is a scaffold product, right? In every sense of the word. Cool. Well, we're very excited about this, and I guess we're going to do a Q&A. Oh,
Something first?
One more?
Okay,
All right.
not a talk. Well, sort of a talk. I want to introduce Jacob Lippincott, who is our new support specialist, and a lot of you have probably already talked to him on the phone or interacted with him through Zendesk, our new support system. And he's going to just talk to you really briefly, and then we would like to use this time first, before the Q&A session. We want to give you a chance to get up, maybe get more food, stretch your legs, and do our user feedback portion of the meeting. So I'll let Jacob take that over. Then we're going to do a Q&A where you can ask us anything you want, so don't go away after you, okay? If you have questions, please.
Okay, so Jacob's going to go through.
Jacob's
Perfect.
going to do
Okay.
it first. And then we'll set up
Okay, so we spent a lot of time talking about features that have already been implemented in our products. So now we're going to switch a little bit and talk about potential features that could be implemented in the future. As said, my name is Jacob. I am in the support department. at Proteome Software, and I have a very short presentation on support updates and user feedback. So first, support office updates. One of the big projects that we've been working on in my side of the building is our new help center. And what we've been trying to do is streamline the support process. And one of the big things that we've been doing is sort of looking at some of our old documentation, modernizing it for the newer version of products, and then migrating it over to this new help center that we're calling it. It's part of our Zendesk, which if you've sent a support request in or anything like that, we've probably communicated through Zendesk, so you should probably be familiar with it, but it's laid out very intuitively. There's a lot of information about sort of FAQs, there's white papers, presentations, a lot of things that's easily downloadable for reference later. Some of the things that are in our programs are very complicated, and oftentimes it's good to have a document written out that gives it step-by-step that you can refer to later as necessary. So, as part of our new help center, you can browse all of our documentation, as I said, view posters and journal citations, and submit a support request. So again, as I said, what we're trying to do is we're really trying to make this a one-stop center for support, so you don't have to go chasing feature requests or things like that all over the web. And if you've seen our newsletter, we are always looking for articles related to Proteome software products. So if you've published recently using one of them, if you send us the details, we would love to add you to our citation list. And especially if you have one here at ASMS this year, we would love to know about it. So let's get into a couple slides about customer feedback, because it's very important to us here at Proteome Software. We are a software company, not necessarily a research facility. So us sitting around thinking about what features we would like to add to our products and what features people out in the field would like to see in our products aren't necessarily the same thing. So how do we figure out what you all are more interested in? Customer feedback. So it is very helpful, as I said, in deciding what features to add to our products. So we get this through email, we get this through phone, or we get this through face-to-face meetings such as ASMS. And again, please visit us at booth 218 for the duration of the conference. I'll say it one more time. And just sort of one more plug from my support office, or the support office, not just mine, is that we are often the bridge between the users and the development team. So that being said, we're going to get into DOTS, a user group tradition. For those of you who have been here before, you've probably seen it. Back in the back corner over there, there's a couple different posters. There's one for each of our products. So we have one for Scaffold, Scaffold Q+, and Scaffold Q+S. There's one for Perspectives, there's one for Scaffold PTM, and there's one for Elements for Metabolomics. You may have also noticed that in the little packet that you got, there are some sticky dots, and there are also some post-it notes. So, place a dot under the heading of potential product features you are most interested in. This is a good way to gain as much information as we possibly can. And then you'll see on the right-hand side there's an additional features section. You can take one of those post-it notes, jot down an idea that's not mentioned, and put it on the additional features section. Or if you would like to provide more feedback about something that's already listed, you could do that on one of those post-it notes too. You also saw a users group survey, if you wouldn't mind taking a few minutes to fill that out. You'll see on the bottom there, it's got a contact section, so if you're interested in receiving any feedback or updates on feature requests or anything like that, please write down some contact information and we'll make sure to get that to you. And one last thing, as it's been stated, we're interested in some more particular feedback, especially in the field of DIA proteomics and in Scaffold PTM related to neutral loss modifications. So if anybody has some experience or information that they would like to share with us regarding those things, please, again, provide us with contact information and we'll be happy to set up a meeting or something along those lines. So that's all that I have. It was pretty short, so now we can get up and stretch.
Okay, go vote and then come back with all your questions and we'll do our best to answer them.
I don't know. I don't use Scaffold, so you know what you want.
I'll do it to Jake. Yeah, you get to.
Okay.
You're my proxy.
This thing